Welcome to CIC bioGUNE, a research institute in the north of Spain where we have 7 magnets dedicated to biological applications and precision medicine in particular. We are very committed with the use of NMR spectroscopy as an application to investigate metabolomics. Metabolomics is actually the characterization of the metabolites in biological fluids. The main hypothesis is that its analysis can provide useful information for the diagnosis of different diseases. This magnet here is a 1 GHz, our latest acquisition and the largest magnet that we have in the laboratory. The GHz spectrometer has a special technology, made HTS semiconductors that allow you to obtain a very high field in a much more compact volume. We are now facing a revolution in medicine that goes from conventional medicine to a new way of medicine where it's personalized. It acknowledges that we are all different and it requires information at the molecular level. In this context, precision medicine metabolism is key in order to understand what are the specific treatments that we have to offer to a given patient. We believe that NMR-based metabolomics will produce a pivotal role to actually generate the new biochemical analysis in the future. We also have here the Fourier 80, a benchtop NMR spectrometer. The new trend of NMR spectroscopy is actually to go to like benchtop machines because they are the only ones that can be deployed in hospitals and clinics and then can be actually run by non-specialists. To that end, what we do here is to try to extrapolate the methodology that we acquire in the 600 spectrometers to these very much more compact machines. The idea in the strategy is to develop technology at higher fields where we can validate with statistical significance and then we can understand the mechanistic value because we have the resolution enough in order to identify the molecules. Yet, in the end, for fingerprinting the different diseases, we don't need that, and all we need is just a validation at a much lower field. We also have here what it would be the IVDR systems. We have 2 of them. One of them dedicated to urine samples and the other one in serum samples. IVDR technology is essential for the proper analysis of metabolomic samples because the standardized operating procedures, the SOPs, require technical characteristics from the sample acquisition and processing that are contemplated in this kind of technology. We have different machines dedicated to biological applications. For instance, this 600 machine with many probes that allows us to obtain new technology, new methodology, new pulse sequences, new experiments for interrogating the matter. Finally, we have this 800 machine that we normally dedicate to investigate protein dynamics and molecular recognition in biological systems. We have, for instance, a score for metabolic syndrome that actually evaluates the cardiometabolic risk of an individual without really taking into account any other anthropometric measurement. On another example, we are actually trying to calculate what is the metabolic age of a person and compare that to a chronological age in a population-based model. The deviations of the metabolic age in the metabolic space can give you a hint on what are the risk factors in the different possible areas, more oncological, neurological, cardiometabolic risk to have, because there are multiple ways of deviating from what it would be the ideal metabolic standard for your age. We have shown you our facilities and our capabilities for NMR spectroscopy applied to metabolomics, and we strongly believe that NMR-based metabolomics will play an instrumental role in this revolution that is happening now in personalized medicine.